Okay, good afternoon, everybody. I'm recording this, so if you want to refer to it later for studying. So today, as promised, we're going to be going over the study guide for tomorrow's exam. Reminder, tomorrow's exam is going to be on cardiovascular, so the 2 lectures on that and the 2 lectures on the respiratory. There will be 50 questions, 2 points each, so 100 points. The exam will be open at 1 and it'll be closed at 5. And you will have 90 minutes to complete the exam just like before. So I want to encourage you before we get started, or recommend that you make sure you do look at the figures we've gone over in lecture. Obviously everything from the hearts, the circulation, to the diving responses, the depth of dives, the durations of diving, some of those, since I will be referring to many of those figures in the exam. All right. So let's go. Okay, so the first topic, obviously, we spoke about was the cardiovascular system. And let's see, make it a little yellow here. Sorry about that. That's not what I wanted to do. There we go. So the cardiovascular system, first thing we spoke about was the characteristics of cardiovascular systems. And of course, whether you're looking at open or closed or whatever animal, they have to have 3 components to it, right? Obviously, some sort of circulating fluid that's going to deliver and remove substances. They have to have tubes that move that fluid through. And of course, they have to have some sort of a pump that's going to create some pressure that's going to move the fluid through the tubes. Having said that, of course, the first thing we spoke about was the differences between what we call open versus closed circulation. Actually, no, hold on 1 second here. Open versus closed circulation. And of course, the difference between that has to do with the fact that even though both those 2 circulations have all 3 components, the difference lies in the fact that with an open circulation, there's not a continuity between the vessels that are carrying blood away from the pump versus ones that are returning it back to. So the fluid that leaves the pump ends up basically leaving the tubes, directly bathing the tissue, and is gradually returned back to the heart. That's nearly not nearly as efficient in our mind as the closed circulation, which is what we have, what all vertebrates have. And of course, that's a circulation that includes continuity between arteries, arterioles, capillaries, venules, and veins that help to maintain a pressure throughout the whole circuit that's going to keep the continuity of fluid going through. I spoke about the pump, and of course, we're most familiar with the pump as the heart. And we talked a little bit about it very briefly, is that the heart is made up of cardiac muscle. And of course, what is the structure of cardiac muscle? You should know the characteristics. Remember that it's a single nucleus branched muscle that has those intercalated discs. Now, when I mentioned the mechanism of contraction, we didn't go in detail over it, but I did mention it's basically the same as skeletal muscle, where you have the release of calcium from the SR that's going to bind to troponin and basically expose the myosin binding site on actin, allowing the cross bridge. The main difference actually is the fact that cardiac muscle is myogenic, whereas skeletal muscle is neurogenic. Skeletal muscle, of course, requires a nerve to release neurotransmitter. Cardiac muscle, remember, doesn't, although it's influenced by the nervous system, the autonomic nervous system, it has the built-in ability to contract on its own with the conduction system that allows electrical impulses to go from one cell to the other through those intercalated discs. We then spoke about the anatomy, specifically of the human heart. Even though it's obviously the most evolutionary advanced, I figured I would just kind of start with it because that's what we're all familiar with. And remember, the human heart consists of 4 chambers. You should know the names of the chambers. You should know the names of the valves. You should know the names of the vessels that go to and from the heart. Now, our heart is part of a dual circuit system where we have a pulmonary circuit where blood goes to and from the lungs, and the systemic circuit where blood goes between the heart and all the other organs in the body. You should know how the blood flows. For example, I might have a question asking you, after blood goes from enters into the left atria, where does it go? What's the next structure it passes into? You could say the left ventricle. Or I might ask you, what's the name of the valve that allows blood to go through the left atrium? That would be, of course, the bicuspid. So you should know the anatomy of the heart, what direction blood's flowing in, and how blood is flowing out. So what you need to know are the chambers of the heart, the valves, names of the valves, and the vessels. I mentioned that the heart is a myogenic organ, meaning that it has the built-in ability to contract on its own. And that's in large part due to the electrical conduction system. We spoke about these specialized groups of cells that are not true muscle cells, but they're also not true neurons, but they spontaneously depolarize. So they do have some nerve properties. And when they spontaneously depolarize, this sends signals to different segments of the heart. What you should know are the names of the different parts, such as the SA node, the AV node, bundle of His, things like that, and the direction, so the order of the electrical signal as it goes from one region of the heart to the other. Why did I say end? That's pretty interesting. I don't think I wanted to say end. Okay, so having established that, then we started jumping into some of the different types of hearts. And even though I mentioned there's such thing as open versus closed circulation, when it came to hearts, we really were focusing on the hearts of open circulation, which would be vertebrates, and we started off with fish. So you should know the characteristics of the fish heart. 2 chambers, 1 atria, 1 ventricle, and you should know the circulation. Now, granted, this is a multiple choice test, so the answers are going to be in front of you, but obviously you should know, for example, that how blood goes from the heart, where it goes, and just very briefly, remember oxygen poor blood leaves the ventricle, goes first to the gills, remember where it gets oxygenated, and then from the gills, of course, then it travels to the other organs of the body, the head and the rest of the organs, where it goes through another set of capillaries. And now this oxygenated blood is now deoxygenated and it returns back to the atria. So you should know that circuit, and you should know the challenges of that, or the drawbacks. Remember, this is a circuit that, because it's a single pump, it doesn't have a lot of pressure. So, and especially going through 2 sets of capillary, 2 main sets of capillaries, the first set at the gills and the second set at the other organs, the blood slows down quite a bit. And getting that blood back to the heart is challenging. But that's where I mentioned, of course, it's the use of the tail, the swimming, that helps to sort of pump blood back, kind of like a skeletal muscle pump. So you know the different characteristics of the fish heart, the benefits, of course, and the drawbacks. We then spoke about the frog heart. And of course, the frog being an amphibian, we're looking at not just living in the water, but living, part in water, part on land. So the structures that may have been beneficial living in the water are no longer applicable, especially in the adult. Remember, as a tadpole, we still have the larval frog still has the gills, but then when it becomes an adult, right, it develops these two other organs, which would be the gills and the skin. So you should know the properties of these. For example, what's important about the heart is now we're starting to see the evolution of a dual circuit. Remember, it's a three-chambered heart. We have the systemic circuit, and we have the pulmocutaneous circuit. You should know where the oxygen-rich blood is going, where the oxygen-poor blood is going, and how it returns. And remember, I mentioned that with two atria and a single ventricle, the concern about the oxygen-poor and oxygen-rich blood mixing, which kind of can hinder the delivery of oxygen-rich blood to the systemic circuit. But remember, I mentioned we've got this spiral valve that helps to divert oxygen-poor blood from oxygen-rich blood. One other thing I mentioned about the frog heart and the frog circulation, of course, is remember that with those different respiratory organs, the role that they play in terms of are they more involved in the uptake of oxygen or in the elimination of CO2. So you should remember that graph that I showed you as far as the relative role of the skin in eliminating CO2 versus the uptake of oxygen, and the lungs and vice versa. Single circuit versus double circuit, of course, that's just a general thing as far as with these cardiovascular systems. Remember, with the single circuit, where do we find that? That's a true fish heart. Whereas with the frog, we're starting to see the evolution of a double circuit where it's basically a double pump. Pump that goes into the systemic circuit and a pump that goes into the pulmocutaneous circuit. But it's not a complete double circuit, right? Because we only have one ventricle. It's not completely separated like it is in four-chambered hearts. Crocodilian hearts. So crocodilian hearts, this is where we first see the true four-chambered heart. Right, two atria and two ventricles. So in many cases, it's like us. But then if we think of adaptations, for example, with fish, we're looking at adaptations for living in the water, the need to extract enough oxygen to survive. We need these really effective, this counter current, right, with the gills. With the frog heart, of course, we're looking at adapting to life on land. Crocodilian heart, one of the things we're dealing with is we're looking with an animal that has the ability to dive, right? So even though we have a double circuit, there's not a complete separation in terms of the vessels. And if you remember, instead of just having the pulmonary artery leaving from the right ventricle and the aorta leaving from the left ventricle, we actually have two aortas. One of the aortas does leave from the right ventricle. Remember, those two aortas are connected by a foramen of Panizza. And one of the things that I mentioned, of course, is that this interesting type of system adjusts when the crocodile or crocodilian is diving. On land, basically, we just primarily have blood going through the pulmonary circuit, through the pulmonary artery. With the systemic circuit, we have blood going, or oxygenated blood going through both aortas to different parts. So why do we have two aortas? Basically, those two aortas deliver blood, oxygenated blood, to different regions. One to the head and neck area, upper bodies, the other one to the lower. What happens with diving is that we don't need the lungs, so the access, or those valves, the cog valves that are normally open to allow blood into the lungs, close, and blood is then diverted into the aorta, where of course it is carrying some oxygen-poor blood, but it does increase the blood supply into the systemic circuit because we don't really need any in the pulmonary circuit. So that's one adaptation, right? This ability to divert blood away from the lungs. The other aspect too is the fact that when it's diving, of course, it's not using its lungs. Fortunately, what happens when the animal is diving, the hemoglobin gives up oxygen much more readily due to the buildup of CO2 and bicarbonate. That affects the affinity. The hemoglobin releases the oxygen, so it delivers it much better when it's underwater. And that's on some of the slides, so hopefully you guys remember that. Then we jumped into the mammalian and avian hearts, and remember, of course, the mammalian and avian circuit are essentially identical in structure and in function. We do see, of course, a difference when it comes to breathing organs, but they're very, very similar in terms of hearts. That was really the first lecture that we spoke about with the cardiovascular system. Second lecture we spoke about primarily the heart and some of the blood. In the first one, the second one, the focus was on blood vessels. So there we learned the anatomy of the different blood vessels, their histological components, arteries, veins, as far as the layers. We learned a bit about their function. Remember, arteries and arterioles carry blood away from the heart, whereas arterioles in particular, even though they carry blood away from the heart, they have a high percentage of muscle that allows them to constrict and dilate, which effectively allows a regulation of blood flow, regulation of blood pressure, whereas the arteries have more elasticity, right? Because they're accommodating blood pressure as the blood flows from the heart. That allows them to expand and recoil. Whereas arterioles, the ability to constrict allows it to divert blood to different organs. This is one of the benefits of having a closed circuit. We then jumped into capillaries. And remember, capillaries are the smallest vessel. This is where the exchange occurs of gases, respiratory gases, as well as waste and nutrients. Remember, with capillaries, there's 3 types of capillaries that really vary depending upon the space between the single layer, the tunica intima, space between those cells. We spoke about continuous capillaries, right, versus fenestrated capillaries versus discontinuous capillaries. And you should know where you find those different organs, find those different capillaries. The common aspect, of course, is they're all a single layer of flat cells. They are all involved in exchange, but they differ in terms of the porosity, or how porous it is, which determines how much the size of the molecules that can pass through. For example, the blood-brain barrier is where we find continuous capillaries. All right. Speaking of the capillaries, of course, this is where the exchange is occurring. And of course, the uniqueness of the capillaries is that here you've got a blood vessel that has 2 things going on. We're moving some material out of the capillaries into the tissue, and at the same token, we're moving waste material, CO2 and other waste from the tissue back into the capillaries where it can return back to the heart and we can breathe out the CO2. All right. What allows this kind of opposite exchange to occur has to do with the pressure differences. So remember, we spoke about osmotic pressure versus hydrostatic pressure. So you should know, for example, when we speak about the arterial end, which pressure dominates and what's happening. Is fluid going out or is fluid going in? And at the venous end, it's what's going out and what's going in. So you should know kind of how that works, the balance of those pressures. Once blood is passed through the capillaries, of course, it's moving very, very slow, relatively low pressure. We're far away from the heart. And that presents an issue because we need to get blood back from the veins, the venules and the veins back to the heart. And especially in bipeds like us, we're vertical, right? Counteracting gravity. I mean, it's an issue in all animals, but it's more of an issue in the bipeds where you're going against gravity and low pressure. So we need to, what are some of the strategies for ensuring blood returns back to the heart? We call venous return. And we spoke about those three different aspects. We spoke about the valves, the one-way valves in the vein. We spoke about the skeletal muscle pump. And of course, we spoke about the respiratory pump. So you should kind of know a little bit about how those work. All right, so while we're on the topic of pressure, we also spoke about flow, and because it's all related. Blood pressure drives flow, but it's not the only thing that drives flow, right? We did speak about a law known as Puiseuil's law. Remember, this is a law. You should know the equation for that law and what are the different parameters. Remember, it's the delta P times the radius to the fourth power over those other factors. So you should know, if I were to ask you, if we increase the length of a vessel, how would that affect flow? If we decrease the radius, if we increase the pressure gradient. So you should know how, I'm not going to have you calculate numerically per se, but I'm going to ask you what happens if we change certain parameters. So speaking of pressure, obviously pressure is one component in flow. Pressure is one of those, or blood pressure is one of those parameters that we have to constantly regulate, right? We need to follow the homeostatic control. We need to make sure it doesn't get too high, or if the blood pressure does get high, how do we bring it down? If the blood pressure gets too low, how do we bring it up? And we spoke about two main components that can influence blood pressure. One is the autonomic nervous system, and the other one is the kidney. But let's first talk about the autonomic nervous system. Remember, that involves the sympathetic and the parasympathetic systems. So remember, I used the example when we're talking about blood pressure. Remember, we spoke about another name, mean arterial pressure, or MAP, and how MAP is influenced by the input of blood coming into the arteries via the cardiac output, right? The heart's pumping. Greater the cardiac output, the greater the pressure. On the other side of the arteries, we have the arterioles. We have what's called vascular resistance or peripheral resistance. And depending on the diameter of those arterioles will also determine the pressure, right? If the diameter is very large, that means there's less resistance, right? That means the pressure is going to be less. It's going to flow through. If the diameter is constricted, right, that's going to increase pressure. So increase cardiac output, increase resistance by decreasing diameter, they're both going to contribute to an increase in mean arterial pressure. So when we look at the role of the autonomic nervous system, and you could probably pretty well guess, of course, even without going over the notes, autonomic, fight or flight, that seems to be like a system that's going to increase pressure. And it is. If the pressure is too low, sympathetic kicks in. And if you remember that table, we see an increase in cardiac output. right? Cardiac output can be due to an increase in heart rate. And also, not only do we see an increase in cardiac output, we see a increased constriction of the blood vessels, which would be peripheral resistance. So those two factors, cardiac output, peripheral resistance, those are both increased by the sympathetic system to increase blood pressure if the pressure is too low. And conversely, if the pressure gets too high, the parasympathetic system kicks in and basically just does the opposite, inhibits cardiac output and decreases peripheral resistance by causing a vasodilation of the vessels. Last thing we spoke about involved the ability for marine mammals to dive. And I do want you to look at those graphs that I showed you. We spoke about some of the different animals, specifically the Weddell seals, but also there's others that can dive for extended period of time. Although, look at the graph to see where the majority of dives are. The majority of dives are short and shallow, but they can also dive very deep for up to an hour. So take a look at those graphs. Also take a look at the adaptations. So remember, we looked at with diving animals, well, first of all, if they're not using their lungs, right, they need to somehow have available oxygen. So take a look at the different places where oxygen could be. Remember, we spoke about myoglobin, which binds to oxygen in muscle. What's myoglobin like in diving animals? Is it more? Is it less? What are some of the other features? What happens to what happens to heart rate? What happens to blood flow? Just very briefly, remember that there's a restriction in blood flow to the periphery, and it's pretty well contained between the heart, the brain, and the lungs, reducing it to other areas. So the animal is not using up the oxygen as readily. All right, oops. All right, next we went into respiratory system. And of course, it's a nice follow-up to the cardiovascular system because you need the cardiovascular system to deliver oxygen, right? So there's a codependence or an interdependence of each other. So there's some terms I introduced you to. We spoke about internal versus external respiration. And remember, external respiration involves the movement of oxygen from the outside world through the lungs into the blood, right? And circulates through the blood. And then that oxygen is delivered to the tissues. That would be internal. So it's the transport, it's the movement. And of course, I mentioned that the reason why oxygen moves in that direction from the outside world to the lungs, to the blood, to the tissues has to do with diffusion, high to low concentration, which is the same mechanism that CO2 moves the opposite direction, from high to low, going from the tissues out. One of the challenges that animals have, and this is what's allowed for the development of different types of respiratory systems, is the variation in oxygen levels. I showed you that table where we looked at oxygen content in the air, at least at sea level, versus oxygen content in the water at different temperatures versus oxygen content at high altitude. In any of those types of conditions where we're away from sea level, you're going to see a tremendous decrease in the amount of oxygen. So in order for animals to survive, they had to evolve specialized ways to extract oxygen from an environment that doesn't have much, right? So you should know the oxygen levels of different environments. We started focusing on, of course, one of the challenges of aquatic respiration. Remember, with aquatic respiration, a couple of the challenges. First of all, there's not much oxygen. Second of all, water is more viscous than air, so oxygen does not move very quickly. Third, oxygen is not soluble. CO2 is more soluble in water than oxygen is. So there's several challenges in basically being able to extract oxygen so that animal can use it. So we're going to talk a little bit about aquatic respiration. I did briefly mention about respiratory surfaces in general. And this would be, when I talk about a respiratory surface, I'm talking about, otherwise known as the exchange surface, where gases have to go from one environment to the other, whether it's from the air to the blood, or from water to the blood, or from the blood to the tissues. That respiratory exchange surface has to be very thin to allow for rapid diffusion. And it also has to have a very large surface area to allow the maximal amount of gas exchange. So first thing we spoke about was aquatic respiration. And of course, at least when we're talking about vertebrates, right? I did speak about briefly some of the other animals like protozoans. You don't have to answer anything about protozoans or anything with that type of diffusion. We're really just going to talk about vertebrates. I shouldn't say that, because we are going to talk about insects. So primarily with, fish, frogs, mammals, birds, and insects. Those are the ones that we really went over. So let's talk about gills. You should know the anatomy of the respiratory system. And this is, of course, in the fish. You should know, for example, that the gills are made up of lamellae, and it's the lamellae, secondary lamellae, which is kind of a subdivision of the gill. That's the exchange surface. Large surface area, very thin, allows for gas exchange to occur. Now, anytime you're looking at breathing, I mean, we know how we breathe, right? We bring air from the outside, right? We inhale, and ultimately with us, even though we're not going to get into details about it, but basically our thoracic cavity enlarges first, which draws air in. Well, how do you draw water into a fish? Well, there's two main chambers to look at, what we call the buccal chamber, which is like the oral cavity, and the opercular cavity or opercular chamber, which is on the other side of the gills. And it's pressure driven, much like it is in us. During the aquatic breathing cycle, in order for water to go from the outside through the oral cavity, across the gills where the oxygen is pulled out, and then out through the opercular cavity, requires pressure changes. And if you think of what we call the pressure-volume relationship, increased volume decreases pressure. Water or air goes from an area of high pressure to low pressure. So for water to come into the buccal cavity, that cavity has to enlarge, lowers the pressure, water comes in. The opercular cavity then decreases, and the mouth is closed. The buccal cavity shrinks in size, gets smaller. At the same time, the opercular cavity opens up, which allows high pressure to low pressure towards the opercular cavity across the gills. And this is where the exchange is occurring. Now, it's pressure driven as far as the direction of water. But what about the gas exchange? It's dependent upon what we call a countercurrent mechanism, where the movement of water opposes the movement of the blood vessels, of blood flow. This is significant because it allows water at any time. If you think of if it's in opposed or just right next to the water is right next to the blood vessels, at every point along the flow, the concentration of oxygen in the water is going to be greater than it is in the blood. So it's going to steadily be diffusing into the blood, which allows you to build up oxygen. That's one of the benefits of countercurrent. Co-current, that doesn't happen. You end up reaching an equilibrium where you lose eventually the ability for oxygen to diffuse is lost. That's what we call co-current. So countercurrent good, co-current not good as far as gas exchange. So yeah, the anatomy and the physiology of fish breathing. Then we spoke about insects. And of course, now we're talking about a land animal. So instead of having this respiratory structure really exposed to the outside, which is what the gills are, even though they're covered by this flap called the operculum, they're still exposed to the outside. On a land animal, you can't have it exposed to the outside because that respiratory organ will dry out and it'll not be functional. So with the insects, remember they evolved this tracheal system, which is this intricate network of tubes that are open to the outside through spiracles that can open and close. And the insect doesn't have a circulatory system. So the way oxygen is delivered, it's directly delivered to the organs through those tubes. So those tubes literally carry oxygen right to the organs, right to the muscles, which is very efficient, right? But this would not work in a very large insect, which explains why insects never really got that big. Thank God, right? Don't want any gigantic insects. Moving on to the air-breathing vertebrates. So we're looking at frogs. We didn't really talk about reptiles, but it would be frogs, reptiles, birds, and mammals. Basically, this is where we start to see the development of the lungs, another internal structure. And in the case of the frog, we're looking at two, the skin and the lungs, as well as the larval amphibian, which would have the gills. So you should know the appearance of when these different organs are present. I should highlight this a little bit more. There we go. Oops. There we go. So remember with the frog, first there's the gills. When the tadpole undergoes metamorphosis into a frog, we see the development of a respiratory organ. The skin, it's very thin, very well vascularized. It's a very good gas exchange site. And of course, the lungs. This is where we really are going to see the difference in roles of the different respiratory organs, right? Between the skin, even though I mentioned it under this cardiovascular system, this is where we really spoke about, which organ plays a major role in CO2 excretion, which one plays more of a major role in oxygen uptake. Remember, this is also, of course, the three-chambered heart, right? And of course, the fish is the two-chambered heart. Now we're into the animals that have four chambers, and that would be both the birds and mammals. So even though the birds and mammals have virtually the same heart, virtually identical, their lungs are not identical. And if you think about it, well, why is that? Well, birds, depending on the bird, right, some more than others, fly at very high altitudes where there's very little oxygen. So in order to be able to extract oxygen, they need to have a system that's probably maybe not as efficient, but pretty close to the gills. Remember, in water, I think there's like the amount of oxygen is like 7 or 8, versus in the air it's about 50 mils per liter, whereas on sea level it's about 200. But anyway, it's got to be very efficient. So the birds have developed very unique lung, very well developed, that has a couple features that enable it to effectively extract oxygen in a different way than fish. First and foremost, it has as its respiratory organ, respiratory surface, at the parabronchi, which are part of the lung. This is where the gas exchange occurs. If you remember the diagram that I showed you of bird breathing, remember I mentioned that when air first comes in, well, in contrast to us, we breathe in and then air comes back out the same way, right? In the case of fish, as well as, of course, birds, basically it goes in a particular direction in the sense that with birds, the air comes in through the mesobronchi, goes through the bronchi, goes to those air sacs. Remember I mentioned they're like a billow. They expand and then they contract, which forces air across the parabronchi. That's where the gas exchange occurs. And then the air enters, the now oxygen poor blood enters into the other air sacs where they're eventually squeezed out. The key aspect of that is I mentioned that it takes 2 full cycles for a particular volume of air to make it through the system. 2 cycles means that there's 2 opportunities to pull oxygen out of the air, which means it's very efficient. We have 1 shot in and then out, right? With birds, you've got 2 cycles to do that. So it's a different system than the fish, but it's still very effective. They don't have a countercurrent system. They have a cross-current system, which still is very similar. Instead of going in opposite directions, it's perpendicular, but still, in this case, the air is the amount of oxygen at any given point. The air has more than the blood, so there's going to be this constant diffusion of oxygen into the blood. All right. So the parabronchi and everything I mentioned in the bird. Mammals, I didn't really talk about the lungs that much, other than just to say that our lungs are efficient. They are very good. Our site for gas exchange, remember, are the alveoli. Much like all the other gas exchange structures, high surface area, very thin, close to capillaries. So that's the criteria for being a good gas exchange site. All right, something that I mentioned earlier, and I guess it just bears repeating, why does oxygen go in the direction that it does, and why does CO2 go in the direction it does? It has to do with pressure gradients, right? Oxygen is at 100 millimeters of mercury in the lungs. Where the pulmonary artery reaches the lungs, the oxygen pressure is at 40. It's going to diffuse in, right? Bring it back up to 100 to the pulmonary veins, back to the heart. Then you get into the circulation. It's at 100, but then by the time that blood reaches the tissues are at about 40 because the oxygen's getting used up. So there's kind of a steady diffusion, and CO2 is just the same. Numbers are different, but it's still high to low. All right. And that, I think, was the end of the first respiratory lecture. The second one is when we started focusing on transport. So we know that, obviously, we've got the circulation. We know oxygen goes through it. We know CO2. But we need to learn about how does it get from point A to point B. And in the case of oxygen, the principal protein that's responsible for that is hemoglobin. And we spoke about the structure of hemoglobin, so you should know the structure of it. And like I mentioned, it basically transports about 98% of all the oxygen. So if without hemoglobin, or if we're low in hemoglobin, we're very anemic, it's because we're not able to deliver oxygen. Hemoglobin has, remember, each hemoglobin has 4 binding sites. Based on the heme molecule. So 1 heme for each globin. So we've got 4 oxygen that could bind. They don't because we're always dumping some off. But that's the principal mode of transport to the tissues. We examined kind of a physiological way of looking at oxygen transport, and this was what we call the oxyhemoglobin dissociation curve. And one interesting feature of that is, and this kind of makes sense, is that the hemoglobin oxygen affinity, in other words, the binding, the strength of the binding, I mean, you know that through chemistry, varies depending on where where the blood is, what part of the circuit, right? Near the lungs, the hemoglobin and oxygen have a very high affinity, which makes sense. Hemoglobin grabs on the oxygen, holding onto it tightly. It's going to carry through the blood. As it gets near the tissues, especially more metabolically active tissues, but tissues in general, we don't want that hemoglobin to hold on to the oxygen forever. It's got to dump it off. So we see a change in affinity, a decrease in the affinity. And this is what's known as the Bohr effect. So remember, the Bohr effect is characterized by an active tissue. And when you've got a tissue that's very active metabolically, you're generating heat, you're generating hydrogen ions because carbon dioxide is being converted into carbonic acid. So high CO2, low pH, increased temperature. Those all help to uncouple hemoglobin from oxygen. That's what we call the Bohr effect, and it causes a right shift in that oxyhemoglobin dissociation curve. Now, we have also what's known as the Haldane effect. And the Haldane effect basically is, what this does is due to, and this really occurs near the lungs, that the affinity of hemoglobin for CO2 is greatly decreased as oxygen levels increase. So there's a difference in affinity. So this allows CO2 to be released into the lungs. That's the Haldane effect. It uncouples from hemoglobin. Whereas Bohr effect, oxygen uncouples from the hemoglobin at the tissues, right? So two different effects that really show kind of the uniqueness of hemoglobin, how it responds to changes. Another effect that I mentioned that is very common in fish, primarily very muscular fish, like tuna, probably salmon, I'm sure, but also eels. It's called the root effect. And this also increases delivery of oxygen to the tissues, but in a much different way. Whereas the Bohr effect, hold on a second, whereas the Bohr effect increases oxygen delivery to the tissues by uncoupling the hemoglobin from the oxygen, so the affinity is reduced, delivers it. In the case of the root effect, a decrease in pH, which is kind of like we saw up here, affects the ability of oxygen to bind to the heme to begin with, right? So it reduces the oxygen carrying capacity. So say, ideally, you would have 4 oxygen molecules binding to a hemoglobin. With the root effect, maybe you'll have 2. I mean, I'm just throwing it out there. So less oxygen binding the hemoglobin to begin with, that frees up the oxygen to bind to the tissues or to interact with the tissues, right? So these two effects in muscular fish, the root effect and the Bohr effect, greatly increases the oxygen delivery, which is important because, again, these are very, even though we haven't said anything about warm blood and cold blood, we often think of fish as being cold-blooded, but there are certain fish that have the ability to regulate their body temperature, and tuna is one of them, and these large fish, and so they really are dependent upon delivery of oxygen because as their metabolism increases, so does their temperature. All right. We also spoke about the effects of hypoxia. Hypoxia is low oxygen. This can happen if you're climbing high altitudes, maybe other pathological conditions. There's an adaptation to hypoxia involving the metabolic intermediate called DPG, diphosphoglycerate. It's an intermediate in the glycolytic pathway. What happens is this builds up in red blood cells. Now, why does it build up? Well, first of all, red blood cells do not utilize aerobic respiration. They don't have mitochondria. So their generation of ATP is through glycolysis. They produce more DPG in a hypoxic state. And what happens is DPG binds to hemoglobin. much like oxygen does. The more, and this is one of those competitive binding things, the more DPG you have, that's going to compete off, not allow as much oxygen to bind, and that's going to increase the delivery of oxygen, which is what you want or might want if you're hypoxic. But you want to increase the delivery. Now, I mentioned that animals are different. Some animals respond to hypoxia or even high altitude by decreasing the amount of DPG, which increases the hemoglobin oxygen affinity, which will be helpful at the lungs. So this is one of these physiological processes that seemingly opposite responses are both correct. It just depends on the animal and the circumstance. In one case, you're going to have an increase in the ability to capture oxygen from the lungs. In the other case, it's going to make it easier to deliver it. So both are useful, both are effective depending on the animal. All right, we've spoken about oxygen transport. What about CO2 transport? So CO2, obviously we need to get that through the blood. It utilizes a different type of transport mechanism. That would be, even though it does use, CO2 does bind to hemoglobin, but to a much lesser extent, maybe about 20% of the CO2 that's liberated, that's produced in tissues, is transported via binding to hemoglobin. It's what we call carbaminohemoglobin. And before I forget, I did talk about carboxyhemoglobin. Remember, carboxyhemoglobin is what it's called when carbon monoxide binds to hemoglobin. And that's, remember, carbon monoxide binds to hemoglobin basically 250-fold increase in the affinity. So that's going to cause a displacement, which can lead to suffocation. So the 20% of the CO2 binds to hemoglobin. That 10% binds travels just through the plasma, the fluid. The overwhelming majority, about 70% of the CO2, is converted into carbonic acid by carbonic anhydrase. And if you remember carbonic acid dissociates into hydrogen ions and bicarbonate. We don't want hydrogen ions sticking around, right? That's going to make things very acidic. So hemoglobin kind of acts as a buffer to bind to hydrogen ion, pulling them so it's no longer free hydrogen ions. What about the bicarbonate? The bicarbonate ends up exiting the red blood cell, and it's an anion, negatively charged. So anytime, with chemistry, you've got to have this electrical balance. If you're losing a negatively charged ion, you have to somehow gain one. And what you gain is a chloride ion. This is what's called the chloride shift. So bicarbonate out, chloride in. You get the chloride shift. Lastly, we spoke about hemoglobin pathologies. I mentioned, of course, sickle cell. I mentioned beta thalassemia. Those were a couple of the different, so you should know what those are, what they're, what causes them. It's a genetic condition, mutation, and what the complications are. So I believe that is it. So let me go back. Here we got 9 people. So anyone have any questions? Would the model animal for the circulatory system be seals or the avian heart? So when you're looking at the closest to a human model, see, when you're looking at the heart, okay, some of the mammalian heart, seal is a mammal, right? So they're going to have the same heart as us. The bird is going to have the same heart as us. When you're looking at the circulation as the properties or what's the best model, it would be the bird, because the seal is different in the sense that you've got the different amounts of these oxygen binding proteins, the myoglobins. So the avian heart would be much more closer to ours. That's a really, really good question. Thanks, Gina. Any other questions?
Hello.
Hey, Jeff.
So in the fish we know, and like in the, I think it was the bird, there's at least some sort of counter current exchange. Is that
They call it a cross
the
current.
cross current
Yeah.
in bird? But in humans, is it cross current, or is it just like how does that principle work in humans?
Basically, so in humans, so you've got the air that comes into the alveoli, and then it just moves across. I mean, it really hasn't been described because if you think of the blood's going this way, so the way I think of it, it's just a simple diffusion. It's not a real counter current.
Mm-hmm.
Yeah. So it's just
Okay.
basic diffusion.
Okay, thanks.
Mm-hmm. Anyone else?
Hey, Professor.
Hey, Daniel.
Hey, could you explain the Haldane effect one more time for me?
Sure. So let me see if I can pull up that diagram. Let's see. Haldane. That was in the second respiratory lecture, I think. Let's see. Okay, let me go back here for a second. Share a screen. Okay, there we got the Bohr effect. Okay, so you understand the Bohr effect, right?
Correct.
Okay. All right. Okay. So it's really almost like the opposite. Okay. So for example, and the wording might be a little bit confusing. So I'm just going to read it and then we can kind of discuss it. So the Haldane effect is the ability of hemoglobin to carry increased amount of carbon dioxide in the deoxygenated tissue as opposed to the oxygenated. So what I was really focusing on the oxygenated. So if you ignore this deoxygenated for a second, and not that it's not important, but just to ignore it, what it's basically saying is that when the blood is in an oxygenated state, high levels of oxygen, and this is going to occur near the lungs, the ability of hemoglobin to bind to CO2 is greatly reduced. So when the blood is very fully oxygenated, it has a lower affinity for CO2, and this is what you find at the lungs. So if it makes it any easier, you can just kind of ignore, this part. It's just saying really the opposite. As far as, whereas deoxygenated, which means that where there's less oxygen, there's more sites available for CO2 to bind at the active tissue. But I would kind of ignore this for a second because I think you're going to get kind of bogged down with the Bohr effect. It's not really necessary to know this part. I would focus on this latter. And if you think of the Bohr effect is what occurs when the tissues are very active, you've got a buildup of CO2, decrease in pH, which lowers the affinity of the hemoglobin for oxygen, and it allows it to be delivered. At the lungs, right, you've got more oxygen, and the high levels of oxygen reduce the affinity of the hemoglobin for the CO2, so it's more likely to give it up. So in one end, we're looking at the reduction in hemoglobin CO2 affinity at the lungs, and with the Bohr effect, we're looking at a reduction in hemoglobin oxygen affinity at the tissues.
I see.
I don't know if that helps at all.
That did, yeah. And thank you for clarifying that wording there. That's what bothered me.
Yeah, it can be a little confusing. And yeah, I probably should have left this out. Not that this is not, part of the story, but if we just focus on what's happening at the lungs, I think it becomes much more clear.
Yes. Yes. Thank
All
you.
right, sure. So Gina asked me something. In the capillaries, is hydrostatic pressure, hold on a second. Is hydrostatic and osmotic pressure occurring simultaneously? Yes. Yes. And it all they're always there. It just depends how they are relative to each other. The osmotic pressure doesn't really change throughout. It's pretty consistent. But what's going to change is whether the hydrostatic pressure is greater than it or less. At the arterial end, the hydrostatic pressure is greater than the osmotic pressure, so fluid's going to go out into the tissue. At the venous end, the hydrostatic pressure is less, and as a result, fluid's going to be drawn back into the blood by the osmotic pressure. I don't know if Gina is still here, but all right. Any other questions? Let's see. We've got Jeffrey, we've got Gloria, Gabby, Zachary, Daniel. All right.
Hello?
Yeah, Gina, hi.
Hi, I just wanted to make sure, so in the arterial end of the capillaries, when you say like the fluid goes out, it's through diffusion, right?
Well, the correct term, it's called filtration.
Okay.
Because basically it's almost like it's being squeezed across, but yeah, it's kind of going from a high concentration to low. But yes,
Okay.
that's at the arterial end. And at the venous end, it's being absorbed. Because the hydrostatic pressure is low, there's no longer the drive to push fluid out, but instead with all the particles in the blood due to osmotic pressure, it's drawing fluid in from the tissues into the capillaries.
Okay, I see. I had one other question. I know
Sure.
you kind of clarified this before, but I wanted to make sure. So when the alligator dives, it increases in it's increased in myoglobin, right? I just wanted to know for sure.
Okay, so when I spoke earlier, and I think I sent I don't want to call it a disclaimer, but so I mentioned in lecture that, why is it able to stay submerged and everything? And I mentioned when it's diving, I think I mentioned hemoglobin levels go up with crocodiles. But I did some more research, and there's actually some recent data that suggests that it's not due to the amount of hemoglobin. What enables the animal to stay underwater longer is that the hemoglobin, when the animal's diving, there's a buildup of carbon dioxide and bicarbonate in the blood. And what happens, that lowers the hemoglobin oxygen affinity, and that allows more oxygen to be delivered to the tissue. So it's not the amount of hemoglobin that changes. What changes is the hemoglobin oxygen affinity due to the buildup of carbon dioxide and bicarbonate in the blood.
Okay, I see. That part just kept confusing me. I'm sorry.
Oh no, I understand. I mean, stuff comes out. You read stuff, and then I just did a little bit more research, and it's like some recent data that sort of said, well, it's not due to high levels of hemoglobin. It's due to the existing hemoglobin just undergoes changes which allow it to more easily uncouple from oxygen, which allows more oxygen to be delivered. So no, that's fine. That's a great question.
Okay, thank you so much.
You're welcome. All right. Anyone else have any other questions? All right. So I'm going to go ahead and finish putting the test together. So tomorrow at 1 or whenever you choose to start, right? It's going to be open from 1 to 5. Just make sure that you finish, obviously, before 5. And you've got 90 minutes to take it. And that's it. So, all right, I'll see you guys later.
Thank you.
You're welcome.